Are we on? Is this on? All right then. Good evening. Welcome to the 2014 CFRI General Membership Meeting. Your voice, your CFRI. I am Sue Landgraf. Most of you know me. I know most of you. I'm, of course, the ED of Cystic Fibrosis Research Incorporated. I am really delighted that you came to join us tonight. Tonight's meeting will provide members of our community the opportunity to vote on research funding, the election of board members, and changes to CFRI's bylaws, as well as hear updates on our finances, highlights of 2013, and a look at 2014 to date. In addition, we will hear from Dr. Jeffry Wine, the director of the Cystic Fibrosis Research Laboratory over at Stanford, with a very informative presentation. But before beginning, I would like to note that no information presented tonight is intended for a patient's diagnosis or treatment. And as always, we urge you to work together with your healthcare team for medical treatment. Thank you to Genentech and AbbVie for supporting this important evening. Your support covers the expense of the venue and refreshments and the studio we have constructed here, plus any other costs associated with this program. Tonight's program is being produced by your CFRI Live team: Scott Wakefield, Eric Martin, David Suhu, and Mary Convento, who is going to be fielding questions from our online audience. So we are live. Thank you, Daryl. We will take applause. They will take applause. Thank you. They volunteer their time. They are terrific. We are live at Bayside Business Plaza in Mountain View, and we encourage all of you to ask questions, whether you are here in the audience or part of the online community. And for our online community, to ask questions, please log into Ustream or sign up with Facebook. And finally, thank you to our volunteers for helping us with our cross-infection guidelines and helping to sign everybody in tonight. And so, now it is time for our feature presentation. Dr. Jeffry Wine. How much CFTR function do we need? A new method for assessing drug efficacy, presented by Dr. Jeffry Wine. Dr. Wine wears many hats in the CF world and is highly sought after around the world for his experience and expertise, and for many good reasons. He is a renowned academic and CF researcher, receiving his bachelor's degree in psychology from the University of Pittsburgh in 1965, and his Ph.D. in physiological psychology from UCLA in 1971. He was a research and teaching assistant at UCLA from 1966 to 1971, and a postdoctoral fellow at the Department of Biological Sciences at Stanford from 1971 to 1972. And he joined the faculty there in 1972. He is currently professor of psychology, human biology, neurosciences, and pediatrics at Stanford. Among the courses he is teaching at Stanford: cellular neuroscience, ion transport and cell signaling, cognitive neuroscience, brain and behavior, introduction to psychology, and the human genome. As I said before, he runs the CF Research Lab at Stanford, where he carries out basic research designed to ameliorate cystic fibrosis lung disease. He is widely published in the field of CF. And of special interest tonight, Dr. Wine spent 10 years on the CFF Therapeutics Development Advisory Group that advised Vertex on the development of the compounds that the CF community is now talking about and eagerly waiting for. So please help me in welcoming Dr. Wine. You are welcome.
Sue, that was a fabulous introduction. Where's my mother? So let me just make a quick check and see if this works, and it does not, so that's because it's turned off. Okay, great. So this is the title, and by the end of tonight, everybody should know. There's a bright light in my eyes. Will that be there all night? Yes. All right, I asked. No mercy. Third degree, that's called. By the end of the night, everyone here should, I hope, appreciate what these bubbles mean and what they can do for the CF community. Some people in the audience have actually contributed such bubbles, And I have an ulterior motive for coming. I would have come under any circumstances, but once again, I need CFRI's help, and this time what I need is help recruiting people, but strangely, not people with CF, but people who don't have CF. That's going to be a big part of making this, this assay more usable than it presently is. So let me just say some of the people who have helped. I never credit people very much. Okay, so the people in blue here are people who have actually done the work, and include Sarah Maudlin, known to some of you. Jessica Char is now the mainstay and is doing all of the major work. So we'll see if we have trouble as we proceed, because this is not supposed to be here. The other set of people who are crucial have little 2s next to their names, and those are the staff over at the CF Center, because they helped us recruit all of the subjects that we were using for the ivacaftor studies that we'll talk about. Okay? All right. So I'm going to introduce 4 basic concepts. This talk is a bit technical, but I'm supposed to be able to explain technical stuff. So I will do my best, but if I'm not making sense, you should stop me. So this is a real sweat gland over here. It's a long tubular structure in your skin, and this is my schema of what a sweat gland might look like. So if we start at the top here, this will be air. This is a thin layer of oil on the skin, and this is the skin. And this represents a sweat gland that's in the skin and producing some sweat. Now, if you look at this round ball here, that doesn't exist in nature. It's just a way to show, almost like a pie chart, that there are 2 different kinds of sweat produced by this sweat gland. So, you want me to pause while people get settled?
Sorry.
No, that's fine.
I thought I saw a spot.
Right. So this pie chart, you can think of it, illustrates what turned out to be the case after some years of study. Given the way that we stimulate the sweat gland, this proportion of the secretions are produced by a system that is perfectly all right in people with cystic fibrosis. So it doesn't need CFTR to function. So we call that M-sweat for the way that it's stimulated, methylcholine stimulated. It works fine in everybody, in you, me, and somebody with CF. And then there's this little quadrant over here with the blue color, and that requires CFTR. Okay? So that's the first concept I want, that there are 2 kinds of sweat. The second concept is that this ratio is, when expressed as a percent of wild type function, wild type meaning normal people, healthy controls. And that's because everything is expressed as a function of healthy controls. It's really important to know exactly what healthy controls are like. So given our current estimate of how healthy controls sweat, then on average, in a healthy control, the amount of blue sweat, the amount of sweat that's produced requiring CFTR is roughly a quarter, okay? And so we get a ratio of C-sweat, CFTR-dependent blue sweat, to CFTR-independent sweat. And the basic concept 3 is that this ratio, C to M, or C-sweat to M-sweat, is 0 in people with cystic fibrosis. They don't produce any of this C-sweat because they require CFTR to produce it. So right away you can see that there's a tremendous advantage with this system. Each and every sweat gland in our bodies is a perfect little gauge of how much CFTR activity we have. Isn't that really neat? So it just tells us exactly how much we have. And that's the fourth concept. So it's really interesting. I'm ending my career working on the same topic that I started. And Doug's been with me the whole way. I can still remember his comments on this paper. 1987 was when we published this paper. The first author is Julie Beam, who is now Julie Desch. And she's now a subject in the latest stuff that we're doing. So I call that continuity. Some people would say that I don't have a lot of good ideas.
Persistent.
So this is an old method that we used. In the old method, what we did was we stimulated this pretty much the same exact way that we stimulate now. And we had an oil layer, but we didn't use an optical method. We just collected all of the sweat and measured it very carefully. And so we had a total amount of sweat. And then we compared the total amount of sweat produced by the C pathway, the CFTR-dependent, and we compared it to the M pathway. And what we found, and this is kind of cool when you enter a field, and it's your first paper in the field, is we produced a result that nobody believed, okay? Because, why didn't they believe it? Because the carriers in this test produced half as much sweat as the controls. Here's the controls, here's the carriers, and here's the CF. I thought that had to be true because when I saw that there was zero sweating in people with CF, I thought that must mean that CFTR is rate limiting for this test. By the way, I did not discover this zero response. That was discovered by a dermatologist, Kenzo Sato, a brilliant guy who unfortunately died recently. But he was the one who discovered that that this pathway existed and that it was zero in CF. We made the next step to looking at heterozygotes, and lo and behold, the prediction was right. You got this linear range from the median here, here, and here of sweat secretion. And people didn't, why didn't people believe this? Well, if you look at the sweat chloride test, people who are carriers don't have sweat chloride rates of 50, sweat chloride values of 50, right? Their rate is not halfway between the normal and the CF rate. In fact, you can't really tell the difference between a carrier and somebody who's perfectly healthy. So with the sweat chloride test. Yet this test really shows it up nicely. So that was quite interesting. It told us that this test is a very different kind of test. And putting the two tests together, I always thought would be a really smart thing to do. They are complementary, and I'll show you that at the very end of the talk. Okay, but those of you who have done some statistics look at these distributions and say, wow, these guys didn't know what they were doing. I mean, what you really want when you do a test is you want to have all of your targets sort of clustered around the mean. You like to see a nice tight bell-shaped curve. This is really a wide scatter. But each one of these tests, in some cases, were done with as many as 5 separate tests and then averaged. So we were quite convinced that these people are really down here, and these people are really up here. So it's something different about the person, not the sloppiness with which we were doing the experiments. And that was our first clue all these years ago. Something really interesting was going on here. Look where these people are. They're just nudged off. These are carriers. Probably they're still in the room, and because we're all kind of helping one another, right? And yet, look how low they are. Yet they don't have CF. They don't have partial CF as far as we know. They're okay. So that was the first hint that you don't really need much CFTR function to be quite healthy. So what did we want to do with these latest tests? The idea was to take this from the, I know I'm driving you crazy. Well, we all have to earn our keep. So we wanted to take this down. Yes.
Would you mind holding the mic? It comes right off
Oh,
the
I
handle.
get it. I would not mind holding the mic.
Thank you very much.
Stand here. You know the old joke about the lecturer who, whenever he got behind a lectern, the students all looked like this, and when he walked away, they just kind of looked around. And pretty soon he was nailed behind the lecture. It shows that behaviorism works, right? If you reinforce my behavior, I'll never move.
Question,
Yes.
was there any attempt to determine those, because of the wide scattering, whether the parent had one gene and not the other?
I see. So the question is, could some of this scatter be related to the flavor of the CFTR mutation? And I can tell you without giving you a detailed answer, that we're pretty sure that that's not where the difference lies, because, as you know, what's now called the F508 deletion, that's so common that that was widespread in this group, and highs and lows both had this. Okay, so but it's a perfectly good question, and it gets us to really what we want to know. Why is there that spread? Where is that coming from, and how can we think about using that when we're testing CF? So here's the current setup now. Everything is the same except that everything's the same except that instead of just collecting a volume of sweat, we're going to look at the individual sweat bubbles. And we stimulate these two kinds of sweating sequentially. We give a chemical that produces the CFTR independent sweat, the M sweat, and then we give a series of chemicals actually that blocks this response and produces the CFTR dependent sweat. And then we look at the ratio, and that shows subject, again known to many of you. He also helped put this whole thing together. He's in there, and you'll see what we see in the next slide when we look through the video. Okay, now I don't know how to do this because I don't have the control on my camera. I guess I have to go back here, and let's see if this will work.
Explain the black spot first.
Okay, yeah, let me explain that, and then so these are movies, and so that's why I have to go back. But that's a perfect thing to tell you what you're going to be looking at in this movie. The movie's only 45 minutes long, so so this is the surface of the skin. You can see the creases, and right here you can see sort of a brownish tinge. You can hardly see it. That brownish tinge is a freckle. For this assay, one of the things that makes this assay so special is that for each person that comes in, we identify about 50 glands. And those 50 glands are what we study over and over again. So we not only say, we know how well CFTR works in this person, we say, we know how well CFTR works in gland 32, this person, right? To make the registration better and to help the camera focus, we put a spot of ink onto the center of that freckle. We try to get the center. In this case, it's a fairly small freckle. Sometimes they're larger, but that then is the registration that allows us then, and as always, you guys are ahead of me, so you'll see some of this later on. So let's see if
These may not be necessarily the same person.
These are exactly not only are they the same person, they're the same test. And if you're saying, why does this look a little different? When we finish this test, I can't get anything past you guys. When we finish this test, we collect the sweat and the oil, and then we blot the skin, then rinse it thoroughly, dry it, and then apply the second batch of oil. And in that process, some of the ink gets blotted away. Okay, so that spot's always going to look a little lighter in the second picture. Okay. The other thing, before you ask me, the reason the bubbles are blue, CFTR doesn't make your sweat blue, right? The reason these bubbles are wine colored is because we put little specks of dye in the oil. The reason for that is some of the sweat droplets from people who have CF, who have been treated, are so small that it would be very difficult to see them without having a dye staining. So we put a water-soluble dye into the oil. So it just partitions in and floats around as little specks, which those of you up front maybe can see some of them. But it diffuses in and turns it blue. Okay? So water to wine is the... We don't do it in the M-sweat. We spent a long time doing it in the M-sweat, but we got carryover of the dye into the second test, and it's confused us because sometimes, as you will see, the responses in the second thing are really small. Now let's see if this is going to work. Does anybody see a little? Does this still work? Oh, there it is. Okay, great. Okay. Now you'll... Okay, see the sweat, see the little bubbles growing? Okay. So that's what they look like at the end of the test. That in real life, that would have been 10 minutes, because this is a very powerful pathway. Now watch what happens for the C-sweat. This is now in a normal control person, the same person, and there come the bubbles. Okay, so for that takes 30 minutes. So this kind of sweating is, as you could see from the beginning, it's much less important. And before Rob asks me, we don't know what the function of this pathway is in real life. We don't know. I think it's so that we can test for CF, but all right. So here's a setup, and I'll use this to illustrate. So here, this is a different person. There's a freckle. There's a little ink spot. And what we can do is we can label these. So this is one. These are two bubbles that have merged. 2, 3, 4, 5. Just count them that way. We usually just string them around. And then if you go over here, you can look and you can match them up. You can see how we can match them up. And not only can we do that for one test, from going from M-sweat to C-sweat, but we can do that the next time the person comes in, and we have done it now over years. So you can have somebody come in 2 years later and find the same sweat bubbles. So that's a great question. Sometimes, and you might even see it tonight, sometimes a bubble will just not appear in a test. We don't know why, but then it'll come back in another test. One possibility is that sometimes, we have to inject these drugs intradermally, and maybe we're damaging some sweat glands. We just don't know. But certainly, what we can do, if we want to have things perfectly aligned, we can just go through at the end and say which ones were in all of the tests, and we'll just score those. Yeah, Scott.
How much skin is that? Is that a square inch maybe?
No, much smaller than a square inch. So this is a 1 millimeter, okay? So the whole area is about a centimeter squared, but by the time the camera and everything is going down, it's a lot less than that. So I think I forget right offhand. This, by the way, is greatly expanded. So this is not the whole, I just blew this up so you could see the bubbles nice and big. So I try to always put the scale bar in there for that reason. Okay.
Jeff, is the control someone who does not have CF or a carrier, or what's the control again?
So the controls are people who do not have CF. And it's, again, I'm going to take advantage of every question that's asked me. We need more such people. We need siblings of people who have CF but don't themselves have a CF gene. We're also looking to test some heterozygotes, but our main interest is getting as many healthy controls as we can.
What's the formula at the bottom?
Gosh, thank you. So many of you are going to say, well, you know, you told us that this is a lot less than this, right? But when you look at this bubble looks pretty close to that. And the formula tells us that when the diameter of a sphere increases by 2, the volume increases by 8. because it's a cubic function. So the volume is much, much less here than it would appear to be here. That's a great question, and I didn't notice that. Okay.
So these are actually spheres, not circles.
Thanks again. Yeah, messing up. What's so great about the oil, no, these are perfect spheres. So we have actually done the experiment, actually not with sweat, but we did it with submucosal glands, which was the technology that led to this. These are glands, something like sweat glands that are in our lungs. And we actually used a prism to take a view of those from the side to prove that they were perfect spheres. We also did some calculations to say if there was a partial wetting of the surface so that they weren't perfect, how much would that throw us off? And it doesn't throw you off very much until they really would collapse, which doesn't happen in the time intervals that we're using when the sweat rates that we're using. What we do get is we get merging. So we're taking these images every 30 seconds, and you can also watch them live, and you can sometimes see the bubbles jump together. Right? So that was a control. So now this is what happens in a CF subject. Now, at the beginning of the movie, I showed you this is the way you start, but this is at the end of the trial. So that's somebody with CF at the end of the trial. They look the same way as at the beginning of the trial because nothing happens. And I didn't think you'd want to see a movie of nothing happening. Okay. So, but what you can see here, to just make it a little easier, so picked out 5 bubbles there. And now we go over here, and you can see they're not there. Nothing's there, not even the little specks that you see are not correlated with these bubbles. So that's what we call no sweating, and that's the typical case. All right. So this is where things got really, really interesting, and what brought me back into this area was the realization that we are now in the end game of CF. So for the first time now, we've made a lot of progress fixing all of the problems with CF. But now, there are drugs that are going to fix the fundamental problem, okay? And ivacaftor is the first of those. Maybe omecaftor is going to be the second in combination, but there's going to be a third one, and then probably for most people with CF, it'll be fixed. It won't be cured because these drugs are things that you have to take for the rest of your life, but it'll be basically restoring those folks to normal function. Now, what about ivacaftor and G551D?
Sorry to interrupt, but
Yes, no.
I'm sure everybody's thinking this. Are you going to tell us what percentage of the population could be helped by these different combinations at some point in your talk?
I'll tell you right now, the combinations that are being thought of would take care of about 92% of people with cystic fibrosis. And the people that are left out are going to be, right now, the people who are left out are people with certain kind of splice and stop mutations. But there are drugs in the pipeline for them too. They're just not as far along. But the idea that Vertex has is to have maybe one pill that almost anybody with CF would take care of almost everything. Over 90%. Okay, so a lot. All right, now what you're looking at is at the end of these tests. These are again, I've even magnified things more, and the reason I magnified them more is you can see how tiny these sweat bubbles are. So this is what somebody with G551D looks like when they are on ivacaftor. And this is the summary of those results. 3 tests off, 3 tests on, 2 tests off, and 2 tests on. There are a lot of problems with doing this kind of experimentation, but these are the results. Here's the surprising thing. So these bubbles here look, it's hard to tell how big they are. So let's compare them to a healthy control. So these are healthy control bubbles, and now those are the CF bubbles that you saw. And if you remember what we just said about volume, these are tiny. In fact, they're only a few percent of a healthy controls. And yet, look what happens to lung function in G550, these kinds of patients taking this. This is why ivacaftor is considered a blockbuster drug, because it makes this tremendous improvement, a 10% absolute improvement in FEV1. And lots of other things are happening too. You've probably heard people gain weight when they take ivacaftor. And there's some evidence now that when people take it early enough, there's starting to be a decline in the infections of their lungs. So all of that's really good. So big clinical improvement with low CFTR function. But here's the red herring, and this is where we're going to get a little bit technical right now. This test underestimates function. We know that it underestimates function because when you give this test, our blue bubble test, to somebody who is pancreatic sufficient, we know that a person who with cystic fibrosis who is pancreatic sufficient has functional CFTR, because that's why their pancreas is working. Also, their sweat chloride levels on average are lower. Instead of being on average 100, they're on average 80 for that group. So we know that they have functional CFTR, but this test doesn't see it, or it rarely sees it. So why? So we're missing something. So we had to understand how it was possible that we were missing something, and more importantly, could we correct for it? And this is where the engineers in the audience are going to be dissatisfied, and the rest of you are going to be saying, move it on. Let's just move on to the next slide here. But the hypothesis is that the primary sweat, as produced in that part of the gland way down deep in the skin before it travels up to the skin, that's perfect. If we could measure that directly, it would tell us exactly how much the CFTR function is. We could see that in pancreatic sufficient people. But on its way to the surface, we lose some sweat. We have three ways that we could think of, and if you think of other ways, I'd like to know. First of all, there's what we're calling physical capacitance. I have looked through the microscope at a sweat duct as I perfused it, and what you can, and when it's before any sweat enters it, it's collapsed because it's an absorptive organ. So when the fluid goes into it, it's just like blowing up one of these floppy garden hoses. You see it expand. Well, that takes time. It takes volume. So we don't, until that whole thing is filled, we don't see any volume. So we're losing that. Second, although we know that the duct has low water permeability, so it wants to secrete salt, it wants to absorb salt in excess of water. It's not perfect. It can't absorb crystalline salt. It's absorbing very salty water. So there's volume lost via the absorption process, which, by the way, also goes on even in CF, a little bit. And finally, although we do a lot to try to prevent this problem, there is some partitioning from the bubble into the oil. Oil and water don't mix, but in fact, water molecules can partition out of especially little spheres into oil. We water saturate the oil to minimize that. Nevertheless, putting this all together, we were able, through a complicated way that's in the papers, which if you want to read, and I'm happy to go over it with you if you like, we estimated that we're missing 0.023 nanoliters per minute per gland. Now that's a tiny amount of sweat. It's about 2% of the sweat rate of a normal, healthy person. But it is more than the sweat rate of somebody who has pancreatic sufficient CF. So we can't see it. And that was another clue that, boy, we're talking about really low amounts of CFTR function in this gland. So what can we do to correct for that loss? We do two things. First of all, the bigger the gland, the more secretory capacity it has, so that the bigger fraction, not a bigger fraction, it's always the same fraction, but the total volume is, as that gets better and bigger and bigger and bigger, the since this loss is, we think, relatively constant, it becomes a smaller and smaller fraction. So something that we can do in this test and in no other test that uses sweat glands is we can select which glands to look at. So we select the very fastest ones to minimize these losses. And then, having made this loss estimate, we add it back. Engineers are always putting correction factors in all kinds of things, okay? And sometimes it works and sometimes it doesn't. But that's what we have done, and that has to be considered a limitation of the assay. And right now, it's not a surmountable one. We either give up or we put it in there. So that we have put it in there. So here are the results. So this is a complicated slide because what you're seeing here are 6 different cystic fibrosis subjects who have been tested on ivacaftor. And this is showing their response rate when they're on ivacaftor. When we just select the top 12 glands, the highest secreting glands, and estimate relative to wild type how well they're doing, you can see that their secretion rates are as shown here. You look over here for the blue bars, they're like less than 1% here. Then this guy jumps up to 4%, higher still back down here. But then when we add in the correction factor, and it varies, it will have varying effects depending on how fast the glands are secreting and so on. Then we get what we think is the best estimate. This is again the 12 best secreting glands with the correction factor added in. And I'll tell you why we think that we're probably on the right track here. If you look at these 4 subjects, these are G551D subjects, and you can see where they lie. They're going between 2 and 4%, 2 and 4%. These 2 subjects are interesting. This is an R117H 5T subject, and this subject is pancreatic sufficient. And so this subject actually had secretion in 2 or 3 glands that just got above threshold that we could see before ivacaftor. That was very nice. This subject is particularly interesting because this subject had the lowest M-sweat rate, the smallest glands, if you will, of any subject, and yet look how big when the correction factor is added in, and it turns out she has 2 mutations, both of which are responding, we think, to the ivacaftor, giving this large response. Even so, overall here, you can see that the percent, even with all of the fudge factors added in, are pretty low. Now, you always want to have converging evidence, especially when you have something that's counterintuitive, that is, that a tiny amount of CFTR function can produce all of this clinical benefit. In like so many other things, the answers are there in the literature. You just have to be able to figure them out. So here's where, for those of you who don't like this kind of stuff, yeah, it's just going to be hopeless. But I'll do my best. It's actually amazingly simple, in spite of what this said. So this is the CFTR molecule. That's what doesn't work in CF, right? It's just a little molecular valve, as you know, that opens and closes and allows chloride and bicarbonate to go across the membrane. Now, how much CFTR function a cell has is based on how many of these are in the cell. And that number is N. So if you have a lot of CFTR, you have a lot of CFTR function if it works. There are 2 other things that go into how well it works. This is for any ion channel, not just CFTR. So the other one is PO is open probability. So the way channels work, they're not like a normal valve. They don't just open and stay open and then close. That's the way, if I were designing the system, I would do it. But we're down now at the molecular level, so they're always flickering open and closed. When you stimulate them, they flicker open a lot, and they're mostly in the open stage. And When they don't, they're in the closed stage. So that's the probability of being open for CFTR for a healthy person with a fully stimulated CFTR. The probability that it will be open, I don't know what you would guess, but I'll tell you, it's only 0.4. It's open less than half of the time, which suggests that ivacaftor could make a healthy person's CFTR work even better, and that's been shown in the dish. And we're really wanting to do this experiment with these sweat glands. The final thing is when a channel opens and closes, when it's fully open, and it goes fully open, fully closed, nothing in between. When it's fully open, how much can flow through it? And it turns out that CFTR has that's called its conductance, illustrated by gamma, and that's pretty low. Okay, why am I telling you all of this? I'm telling you all this because mutations affect these things differently, and that leads us to predict and how in the way these drugs will work and what we should see with the sweat response. So what G551D does, and the only thing it does, as far as we know, is it reduces the time that this is spent open. So it's almost always closed. All right, so what can we do with that kind of information? So here is a way to cross-check our results with results that were done in dishes to see if they're correct. Let's just make this bar represent the wild type NPO, that is the total amount of CFTR function in somebody with G551D. What is the measured effect of the mutation, G551D? Well, it knocks the open probability down to 1% of wild type. So that's pretty low. Now, ivacaftor will increase the PO, this has been measured in the dishes, to 10 to 30% of wild type. So that's why everybody said, wait a minute, you're getting 10 to 30% of function, so of course you're going to see this big effect on the lung, and you should see a lot more than you do in your sweat. But let's think about this a little bit. First of all, there's two estimates. One's 30% and one's 10%. That's a big difference, all right? For reasons that I won't go into now, we think that this 10% is a better estimate because there are problems with measuring these kinds of POs in channels. But there's another thing that people forget. People with G551D almost always have something like F508 del on the other mutation, which doesn't work at all. So they only have half as much of this function. So that drops them down to 15% or 5% of function. And that's, for biology, pretty close to what we're getting. We agree more with the 5% than with the 15%. And I think the field's starting to come around to this lower view, okay? So this is an independent way of cross-checking and saying, yeah, these low numbers that you're getting are probably real. Why is this important? I'm going to ask you a question. Why is it important?
You can fix the delta F508, I suppose, huh?
Well, exactly. So if you're trying to raise money to start a company to fix CFTR, and somebody says you don't actually have to make it 100%, you make it 10%, you've got a company. That's caught a lot of people's attention, and it's good for us, it's good for the world. So I hope eventually it's 100%, but it doesn't need to be, and it isn't in us, in fact. So what about the R117H5T? You sure can.
So I remember probably you saying almost the same thing 10 years ago before any of this research was done. Just because we knew about the pancreatic sufficiency, insufficiency, mutation difference. And it was basically the same message, that we wouldn't need all that much correction. what have we learned from this that we didn't know before? I'm sorry to put you on the spot.
No,
but
you're
I
not
think,
putting
but
me
I
on the
think we
spot.
have, and I'm just
No, no.
wanting to see it clarified.
You're right. I've been saying this forever, and people have been ignoring it forever. Now we have evidence. That's the only difference. Nobody else thought this was good.
And we have an assay.
Well, that's where the evidence comes from.
Yeah. But I mean, you can use it to test drugs.
Yes. I forgot about that. These guys are helping me out. This fella's not. All right. So having gone through what we just went through with the N and the PO and the gamma, you'll appreciate what happens here. So first of all, R117H reduces the PO, but it doesn't reduce it very much. Instead of G551D, which sends it to the cellar down to 1%, R117H drops the open probability down to something like 30%. So, according to me, if you have 30% function, you shouldn't have CF, right? And in fact, people with R117H don't have CF. Even if they have another allele, F508, on the other thing, they still don't have CF. So so what causes people with R117H to have CF? It's this little 5T sticking out there. Because what 5T does, 5T reduces N. 5T, you don't, it's complicated to explain why, but it causes a lot of misprocessing of CFTR, sort of like the F508. No, it's different than that. Just makes, there's fewer molecules made, and so it drops it down to 10% of what it would be normally. So let's go through that explanation. Here's a wild type. 2 alleles, 100% function. R117H drops the PO down to 15 to 24%. Okay, a little more than I said. All right. Again, in fact, this difference is within the same laboratory, just different methods of measuring. It's not easy to do these experiments. Now the 5T comes along, that drops it down to 10%. I have this 14% here for reasons that are complicated. We are not at 100% of N. We're probably on average more like 70%. And just to jump to the, I'm not going to talk about this too much, but that wide spread, I think, has to do with how many normal CFTR molecules those of us who have them really do have. Now you put the two together, right? You have 15 to 24% of function in only this many CFTR molecules, and now you're down to 2 to 3.4%. But remember, these people have an allele on the other chromosome that's not working at all, so you have to cut that in half. So this is what this kind of analysis tells you somebody with R117H5T has, and these people are pancreatic sufficient. Sweat rate drops to about 80, pancreas works. What does ivacaftor do? In theory, ivacaftor, let's just say, and it hasn't been done carefully in a dish, so if we make the assumption that ivacaftor makes this go back to normal function, then somebody taking, with this mutation, who's taking ivacaftor should just be like somebody who only has a 5T mutation. They should have about 7% wild type function, and what we measured was 6%. So again, that's a pretty good relationship. So who knows? Maybe it's 5% more or less. We don't know. Okay, we're almost finished. This is the traditional sweat chloride assay. It's a logarithmic function of CFTR expression and function. So this is the first time we've been able to actually do this, to say, okay, let's just spread CFTR function out based on the sweat bubble assay, going from 0 to 100 over here, and this representing the wild type, realizing it's an average. you get a linear readout. That's what the heterozygotes tell us. And this is where the sweat chloride values are. This right here represents the sweat chloride values of people, G551D subjects taking ivacaftor, and this is what they measure in the sweat bubble test. Okay, so we're almost finished. So the summary is the benefits are large clinical benefits from low levels of CFTR function relative to no function. Just pointing this out is tremendously encouraging for drug development efforts. And if somebody wants to develop a drug and they get it into humans or want to get it into humans, this bubble assay allows testing of these new drugs in very small cohorts of subjects. And we know the answer right away. As soon as we do a test, we can say whether it's working or not. People, the other thing is that people with normal sweat chloride tests may in fact have quite low CFTR function. We're starting to see such people. And this might have health consequences, like those carriers that I showed you in the original tests. So some of us may have something else going on. What are some limitations of this test? Well, we need correction factors, and the solution to that is to keep constantly refining them, taking more data as it comes in from other kinds of experiments to make them better. The control population is small, and with a few exceptions, it's not genotyped. And this is what you guys have just helped us to do. We want to expand this population. We now have the resource to do it. We just need the people. So if you know anybody who's interested in CF and interested in helping CF but doesn't themselves have CF or even a mutation, we'd love to see them. And thanks to CFRI, we can actually pay them for their trouble. The assays are mostly unblinded. That is, we always knew for most of these subjects whether they were on the drug or not on the drug. And that, scientists just do not like that, right? They just hate it when you actually know what the outcome is supposed to be. Fortunately, we have now started, and there are political reasons why that had to be the case. But now we have done some blinded trials. We're hoping to do a lot of blinded trials, and that will help. We're also going to do automated image analysis. We're hoping to get this automated, so any kind of error will be taken away. But I think you can see it's pretty hard to kind of fudge this test. There's either nothing or there's something there. So far, the testing's only at Stanford, and also scientists hate that. They always want to see replication, as they should. Fortunately, we're now setting up, so if you had to pick any place in the world to set up another test, I couldn't have picked a better place. We're going to set it up in Verona, Italy, right? So two wonderful MDs came from Verona and learned the method. Yeah, that's right. So all my subjects will want to go to Verona to take the test, right? And we don't have quantitative measures of models of sweat gland function, both for secretion and for absorption. And so we really need to get such models. And I think that's the end. This is just the program, and what you guys are really helping me with are these two things here. But we have a variety of other things. We're now starting to do sweat chloride at the same time. And my dream is to really have a little chip that you slap on the arm, and all of this is done with no oil and no dye and nothing, right? And I guess these two guys are going to do that for me. Okay, I think that's it. Yeah. Yes.
Are you looking for non-CF subjects across the age spectrum band and gender, or is there a target population you're looking for?
That's a great question. And we think, we're not certain about this, but we think that the age shouldn't matter because of the ratiometric way that we do this. So for example, when when people are, so what really counts more than age is probably activity level. So if people are sedentary, their sweat rates go way down. But both of them, they go down together, so when we do the ratio, it's supposed to compensate for that. We don't think it does exactly, but the short answer is we'd like to get a lot more younger people than we have right now.
Young adult or younger?
Young adult. So only we're approved to do minors, but we would prefer to do only adults at this stage.
What is kind of the study? So if I ask a friend to do it, so what do we study?
So that's a good question. So what it entails, well, there are two studies that we're trying to get started. So the standard, just the sweat rate study, would mean coming to the laboratory at Stanford for a 1-hour test 3 times. And
In which period of time?
so we don't do it more frequently than once a week. Okay? So the quickest it would be over for somebody would be in 3 weeks. But they can be spaced out in much longer periods than that. There's another aspect of the study, and this is going to take more commitment than the other study, and that is to this question of how much CFTR a normal person has, and we think it's over a wide range. There are methods to measure that directly by measuring the RNA in the cells, the messenger RNA. But to do that, you have to get the cells. And the way to get the cells, the easiest way to get the cells is nasal brushing. So that's more invasive than this. This involves sticking needles in the skin, so some people that's the end of it for them. But they're very tiny needles. They're the same kind of needles that the diabetics use, okay? Very good. And they don't go in deep. But for this nasal brushing, they take a little, it's kind of stiff bristles. I haven't had it done yet. I'm going to be the first volunteer, and I'll let you know then. But what people say is nobody likes anything stuck in their nose, right? It goes up fairly far into the nose, and then they rub it fairly vigorously, and so you're supposed to tear, and you don't like it, but it goes away. But they don't use anesthetic for it, which they could do. They could put a squirt of lidocaine up there, but they don't even bother, so that suggests it's not that bad. But I'll let you know.
So you say, can we carriers do it, or just?
Carriers can also do it. Our key interest right now, well, carriers can do it, yeah, because we also need to expand that set a little bit. But I know what's going to happen, right? Knowing this group, I'm going to get a bunch of carriers and no non-carriers, right? I've talked too long. Thank you very much.
Jeff? Thank you. No, come back. So on behalf of CFRI and our membership and our community, we'd like to present this to you. Now it's an orchid. Why is a guy getting an orchid? Well, it's purple, which is our color. It's tall, like you are. But it's also delicate. It's fragile, and it needs tender, loving care. It needs to be put in the proper place, given the proper nourishment, to be taken care of.
You've been talking to Marlene.
No, this is something that a scientist and a researcher can do. And so this is for you, and thank you very, very, very much.
Thank you.
Now, I think we need to do some switching out here, some technical rearranging, as it were. And that was fascinating. And I think you're right, Jeff. I think you'll get everybody in this room knocking at your door. There you go. Okay, now we will go on to the general membership meeting. There's our agenda.